Hello, everyone. Welcome to the Thomas Expert Series, and thank you for joining us today. We're going to get started in just a moment. In the meantime, please double check your microphones and make sure you are muted. This Expert Series will be recorded and placed on demand within 24 hours, so please let your colleagues know how much you enjoyed the presentation today, and you can direct them to the On Demand tab or have them sign up for future talks. To get everyone a little familiar with the chat, I want to see where everyone's tuning in from today. I'm in New Jersey. Gavin, Maria, where are you guys at?
I'm in California, Irvine, California.
And I'm in Solon, Ohio.
And we've got Philly, Pittsburgh, Florida, North Carolina in the chat, Georgia, Ohio. Awesome. Got a nice mix in here today. Another North Carolina, a couple more North Carolina.
A lot of from North Carolina.
And in New Jersey. All right, I'll go ahead and get started. So at this time, I'm going to go ahead and turn it over to Maria and Gavin here today to talk about sample preparation solutions for environmental and agricultural research.
Thank you, Heather, and good morning, afternoon, everyone, and welcome to this episode of the Expert Series. So my name is Maria Burton. I'm joined today with my colleague, Gavin Wang. He's our instrument specialist. And this presentation will be a joint effort. And the title of our talk today is the Sample Preparation Solutions for Environmental and Agricultural Research. And we're going to be talking about what MP Biomedicals can bring to researchers that are actually using this type of samples.
Yes.
I see.
Or
Okay.
Maria, or I can try and share my screen.
Let's
yeah,
do
why
that.
don't
I don't know what is going on.
Yeah, let's do that because I can't coordinate these two screens together. Thank you, Gavin. So while you're getting that up and you're seeing Meet Our Specialist, so again, my name is Maria Burton. I'm the Product Development Manager for Molecular Bio. And as soon as the presentation comes up and we can share the next slide, one of the topics I will be discussing today is the sample preparation solutions we have using our fast prep systems. And before we delve into the solutions, of course, we need to understand what sample prep is all about, what are the current challenges so that we can see what the solutions are, and of course, how we are able to provide complete solution from sample prep to molecular analysis in a way that guarantees more successful experiments and analysis of DNA or RNA or any target molecules. So what is sample prep? And sample preparation is a process by which we take biological samples, we treat them with the hope that we can release most of the target molecules that most researchers want to analyze, such as DNA, RNA, proteins, or metabolites, and make them available or in solution so that they can be used for purification and then be later on applied in downstream analysis. Most of you know there are quite a few disruption methods that have been developed over the years and they're available commercially. Different categories apply. First, mechanical lysis. As the name implies, this type of homogenization or tissue disruption takes advantage of force, and then by applying force to the tissue or the cells via a mortar, pestle, downs, blender, or a rotator or a stator equipment, as well as a bead beating or the beater method, we are actually inducing this type of disruption in the tissue and then actually opening up cells and being able to release the material. This type of disruption can also be achieved with pressure, as in the case of the French press, also by temperature treatments, freezing samples and thawing them quickly. It's a good way to burst cells open, as well as using heat. There's also the use of sound or sound waves to achieve the same type of disruption. And as you know, there's also most of the extraction kits take advantage of chemical enzymatic lysis, where they use, is that out of the way? Sorry. Where they use lysis buffers and detergents, and then create this osmotic pressure outside the cell, using hypotonic solutions to be able to induce this burst, and then be able to release or be able to extract those molecules. Next. Since the methods are quite different and they use different types of means to induce rupture, you can see that this could bring challenges in sample preparation. And the challenges that we see for these methods mostly come from three categories: sample quality, contamination and impurities, and adaptability. And I'll go through some of the pain points here. Sample quality is compromised when the homogenization doesn't proceed completely, is not done right, and therefore most of the material is still trapped within the cell and leads to low yields or target molecule. Manual methods, as you know, can be challenging as they are less reproducible, can be inconsistent, and also take time. And some of these methods that are manual, like the French press, usually are very heavy and kind of hard to manipulate and apply pressure. Also, depending on how the cells are lysed and the tissue is processed, it can lead to increased impurities and also damage your target molecules and impact their integrity. Sample diversity is also an issue here, as each sample when we extract, these methods are not usually adapted to that type of sample, and then some may be better for certain tissues or versus other type of material. Contamination also is a challenge that comes in sample prep, depending on the type of method that is being used. Open homogenization methods usually tend to bring in environmental contaminants versus a closed system where there's no chance of bringing in this type of environmental impurities. Methods that also use chemicals or enzymes can interfere downstream, whether you are doing an PCR or any type of amplification reaction. Having this type of chemicals can actually interfere with the activity of these enzymes. Also, there are impurities that are inherent to the type of sample that you're working with. Today, we're featuring mostly the plants and soil. As you know, plants have a high content of carbohydrates and polysaccharides. It's a long chain carbohydrates and polyphenolic compounds, as well as DNases that can compromise your target molecule. And soil, of course, come with other impurities such as humic acid, heavy metals, and other impurities that can also, if it's not removed after homogenization, can compromise the downstream analysis. And the other challenge that some of these methods have is being able to adapt to the different needs of your sample. For example, some are best for soft versus hard tissues or hard materials. There's a challenge of sample variability. As you know, soil, fecal matter, and things like that tend to have inherent variabilities, and be able to handle them in a way that can guarantee that the most DNA or target molecule from them. Also, the way we extract DNA with these methods can generate heat that can compromise the integrity of your molecule. So working under cryogenic conditions tend to be better to preserve them. And there's also a challenge of handling too many samples, or sometimes a few samples is okay, but when the throughput increases, it's important to have options that can handle low to high. and can be reproducible, whether you're working with 2 to 10 samples versus more than 20, for example. And the ability to automate is always desirable when we're talking about sample prep. Next. So this is where we feel our fast prep systems can meet a lot of these challenges that people have with sample prep. The fast prep system is actually a bead beating technology, so uses the mechanical disruption and lysis to achieve homogenization and lysis and preparation of your sample for extraction. These instruments are actually not only come with a shaking and providing that mechanical, that movement, but also contain certain, provided with tubes, what we call lysing matrix beads. And these beads are designed together in a closed system in a tube or a vial to be able to more efficiently lyse your samples by colliding with the material. Once they are adapted into the machine, that movement can actually achieve this type of homogenization because you have the impact of the beads on top of the shaking of the instrument. And the fact that we also have adapted or designed different types of beads that can optimize lysis for different sample types. So as you can see here, you can see metal beads for harder material versus something a little bit more softer for tissues like the lysing matrix D. And the image also shows the different types of throughputs at the fast prep as we have developed with the fast prep system from very low to high, but Gavin will talk more about this. Next. So more on the advantages of a fast prep homogenization or bead beating system. Again, it's, as you can see, it is a quick and easy and convenient way of doing it. We said quick because the cycles or the time needed to disrupt your tissues are usually between 40 seconds to a minute. It's easy because already with the vials and beads that we provide, and they're sterile, so they will not add additional contaminants to your prep. You can just load your sample, close it, and it's just place it on the adapters and run the programs. The mechanical lysis does not require chemicals or enzymes, although this could be used as part of the lysis and the lysis buffer to remove additional protein out of your preps, and therefore you have less interference for downstream processing. The fast prep we have seen maximizes yields as you are actually being able to thoroughly open up your tissue, your cells, and recover intact molecules. It's also consistent. This method is also amenable for high reproducibility and reduced variability. And why we say that is because once you load your samples across in your adapter, each tube receives the same type of shaking and force and then homogenization that across the ring. So there's no variability in the actual process, as well as this could be repeated many times and with really high reproducibility. As being a closed system, again, as I mentioned, it really minimizes the cross-contamination or environmental contaminants during homogenization, and it can be tunable. As we mentioned, the types of beads together with the different programs that we have optimized in the fast prep are actually being tested and validated for specific sample types to improve this process. And it's flexible because we do have different adapters, whether you're working with small volumes like less than 2 ml, or you need to scale up volumes to up to 50 ml. There are tubes that are available with the beads, different sizes. And of course, as you can see, it could be a higher throughput as you get to know the different fast prep instruments we have. And Gavin will talk to you more about the solutions we have for cryogenic capabilities as well. Next. And so we feel like MP Bio can provide this complete sample preparation workflow and optimize it from the start. As you know, things don't start right, don't end right. So if we have, by preserving first, having good samples, fresh samples to start with, and then using the homogenization solutions that can best slice your tissue according to the strength, the softness, the type of sample that you're dealing with, that already optimizes this workflow. And then we provide solutions as well, and Gavin will talk about it, extraction kits that can both extract DNA, RNA, protein from in a manual format, also in an automated way, and that will be coming soon, and that can provide really high purity and yields. And of course, once you have good quality material, is key for your downstream analysis so that you can get the best data, whether you're amplifying your gene, detecting SNPs, or sequencing, that you have quality material, enough material to actually go and analyze your molecules. I think this is where my slides end, and now I'm going to turn it over to Gavin, who will then talk to you more about the actual instruments and how you can benefit from the different formats that we offer.
All right, thank you so much, Maria. And I will take it here to actually show you a more specific on the instruments side of what we are offering. So as mentioned by Maria, we do offer with our customers, depends on your daily needs or your sample types. We do want to cover SMP. When we are offering this solution to the customers, we offer a wider variety of surplus as well as a wider variety of sample types. So we are fitting your personal workflows and your researches. So starting, we do have the Super Fast Prep 2 series, which is more like a hands-on. You can bring this instrument to your field if you want to process that right at the location where you collected your samples. This is a handheld homogenizer which can process 1 or 2 samples. Each sample, the map was the 2 ml tubes that we offered. This is the size of this is really small. You can handheld it, carry into the field, and actually homogenize your samples over there. In terms of a slightly higher throughput, we do offer the FastPrep 5G series, which is our 24 to 48 throughputs. This instrument is one of our best selling products. So this fits your need if you are filling. You do have multiple samples anywhere. The adapter fits 24 samples. Of course, it can be less than 24, but anywhere between that is where you will consider the 5G, which is a mid-throughput homogenizer. This instrument has the capability of interchange with different adapters. So, for example, if you're not if your sample type is exceeding 2 mLs per sample, for example, a lot of cases for like wastewaters or larger plants, if they are exceeding 2 mLs, we do have the options to upgrade to 4.5 ml tubes, 15 ml tubes, as well as 50 ml tubes. So this instrument had different adapters that can interchange to fit your sample volume needs. And we also have our highest solution, which is the FastPrep 96, which is you can process up to 192 samples per run. This is mostly used by our customers who are dealing with agricultural studies with multiple samples, and then they want to perform high throughput homogenization at the same time. So the instrument has the capability of adapting 96 2 ml tubes or 2 depot plate, which can allows you to process up to 192 samples at a time. These have interchangeability for different sample types as listed on the right side of the slide. And for the there's also protocols online with being pre-optimized for your particular tissue type, plant type, or soil with water, for the samples that the parameters that you'll be set. Our 5G instrument is also built into the instruments where you can just select your desired samples, and then there's a pre-optimized program in there. In terms of the time of the instrument running, as mentioned by Maria, most of the times only requires 40 seconds to 60 seconds to process your sample, with exceptions of super hard sample, usually like seeds. So if you are processing like soybeans, these might be taking a little longer just because of the nature of the sample. It's much harder compared to other samples like leaf, wastewaters, or soils. So for these type of samples, they usually require 3 minutes to process, so three runs. But for most of the samples, they are covered in the range of 40 seconds to 60 seconds. In terms of the, so that was more focused on the throughput-wise. In terms of the samples, we do offer a wide variety of lysine matrix, which is, and showing you on the slide is a guidance to showing you, depends on the sample type, which type of lysine matrix you can choose from us. We do offer a wide variety of lysine matrix, so we have a category of showing, depends on the hardness of your sample, we do offer different solutions for you. Because one thing we want to, depends on the downstream of the workflows. While the homogenization is in progress, we also want to make sure that your sample is not completely degraded in that process. So if you choose a very aggressive lysine matrix with a higher running time to treat just a soft tissue, that may cause the issue of degradations. So we do have solutions for you. There's also plenty of citations online to see how the colleagues in the field is also processing, or other researchers is processing their sample with our lysine matrix. But we do have a wide variety of solutions that can be tested out. There's also recommended lysine matrix built into the instruments where you can take a look to see what your sample, what lysine matrix fits your sample the best. So they come with different combinations. There is less aggressive and more aggressive, depends on which lysine matrix you will be choosing. But no matter which type of samples that you are processing, we'll have a lysine matrix for you to process your sample. Here's just another representation of the lysine matrix that we offered. Here is a very general guidance on what, based on all the customer feedback that we collected, of our recommendations of what type of lysine matrix depends on the sample that is listed above. And then what is most recommended is A, B, C, D, E, M, Y, or Z. In terms of the sizes of the tube, as I mentioned, we do offer for all lysine matrix, we do offer the flexibility of the 2 ml tube, 4.5 ml tube, 15 ml tube, 15 ml tube, or the 96 wall plate format. Each of these, all of the tube are pre-filled, so you don't need to worry about filling the tubes yourself or a cross-contamination happen when you are filling the tube. All these are sterilized, pre-filled tubes where you will just need to throw the samples into the tube prior to homogenizing our instrument. Aside from the tube that we are offering, we do also offer the downstream workflows after your homogenization of your samples. So we do offer a wide variety of kits that associated with your downstream workflow, especially for your nucleic acid extractions. So after you process your using your fast prep instrument to homogenize the sample, break the cell walls, release the DNA and RNA out, we do have the specific kits that is associated with that. So our kit is also famous when we're comparing to competitor for its high use of DNA, RNA from plants, environmental samples, and the tissues. We also have the feature of removing polysaccharides, polyethanols, and other inhibitors during our during the design process for our kits. Listed here is the two famous kits that we have, which is the Fast DNA Spin Kit for your soil research. There's also going to be the Spin Easy Kit series that we are launching. So that is also something that you can check it out. And then the nucleic acid being extracted from the kits will be readily available for your downstream applications, no matter it's PCR, qPCR, NGS, and other downstream workflows. If you are also considering other automated solutions, we do also offer a automated nucleic acid extraction system, which is our Metaplex system. This is a magnetic bead-based nucleic acid extraction. It uses magnetic rod to extract to assist your automated workflows of extracting DNA and RNAs. So this is a high throughput and flexibility is what this instrument is known of. So it can process 1 to 48 samples individually, which means that you can, no matter if it's one sample, we do offer a single stripe of the kits. So you can just use that one stripe to extract all for a single sample extraction, and that flexibility offers from one sample all the way to 48 samples. But this instrument is built to, you can process up to 96 samples per run with the pre-filled extraction kits. So we do have the pre-filled extraction kits. They are, all you need to do is, they're individually sealed. So all you need to do is pull off the seal on the top, adding your samples, even either processed by fast prep or from your other homogenization method, as some samples does not require a sample prep step. It just needs to be added in there. And then it's a highly expandable platforms. We currently are offering the four types of kits, which is the animal tissue, soil, plants, and stew samples, where you can process in this automated system. We have checked it do also provide a high reproducibility, and it do comes with the features we can simultaneously extract the DNAs or RNAs. So once it depends, again, the only differentiation during that extraction process will be if you want to perform the DNA extraction, you will be adding the RNAs, versus if you want to do the RNA, it will be more the DNAs that will be added on the kits. But the kit itself provides you the flexibility of either performing DNA or RNA. They are designing one pre-filled kit which you can easily extract from the instrument. The average running time for the application above to process 96 samples is 35 minutes per run. And this instrument is our newly launched instrument. So if there's any interest, please let your rep at the Thomas Scientific or us know. Again, so we do have all kinds of publications online that you can also be checking it out for the specific workflows that you are interested. But we do have all kinds of flyers that shows our reproducibility and the advantage of using the fast prep solution plus in our no matter automated or manual extraction method. So these are usually can be found on the website. Here is just a solution that we showcase for the sample preparation solution for the wastewater analysis using the fast prep. You may be asking that why do I need to homogenize wastewater when we are? It's just how can we possibly homogenize a liquid format? So in terms of the homogenization, a lot of times when we are adding when we're trying to analyze the contaminated materials in the wastewater, we are trying to analyze the bacteria or the viruses that is contaminating the water. While using the fast prep, our lysine matrix during the homogenization of the wastewater liquids, and it has the capability of breaking these cell walls, where you can then use either FastDNA or Spin Easy series of product that we offered for your downstream extractions. So these are is being proven. You can check out more. If there's any questions, feel free to ask us on the website. And here is the a new product, as we are newly launched our Spin Easy kits for the, as I mentioned, the previous workflows for the wastewater. The kits also come with the filter to collect the sediments and concentrated microbial content for DNA extractions. So this will be, this is something that we offer for advancing your research related to the wastewater and the actual content in the wastewater for your research. And this improves the efficiency as well as the actual results of your experiment. So if you are ever interested in the wastewater for the in terms of the extraction, we do offer these solutions for you. Our kits always come with the options with along with the lysine matrix or without the lysine matrix version. So a clarification to make is you don't for the downstream extraction kits that we are offering, it doesn't require you to purchase a fast prep in order to use our downstream application kits to perform the extraction. But we do recommend, as these is the most validated protocols that we run, and all the protocols we validated, that this will provide you with the best results for your extraction and sample prep steps. Again, as we are focusing on the environmental and the agricultural research today, I'll only use, and due to the time limitation, I only use the example for the wastewaters, but there's also other case study related to the fast prep system to use agriculture and environmental sample that you can be checking it out for, for tobacco leaves, for leaf and roots, stem milk, and agricultural soil studies, qPCRs, metabolism profiling, and quantitative genomics. These are all available at the website where you can check it out for these different applications that is using the fast prep. As this is a long leading homogenizer on the market, I'm sure we can help you, assist you finding your sample type and how the researchers in your field is using the fast prep system to help giving a much higher reproducibility and a consistency for their research. All right, so that will be the presentation for today. If there's any questions related to the system, feel free to ask me or Maria, and we can jump into the Q&A sessions.
While everyone is thinking and typing out their questions, I'm going to go ahead and share my screen for just a moment.
Yep.
Okay. So in here, you will see that MP has a giveaway if you take the survey that is in here. There's also literature, including the piece that Gavin had spoken about in his presentation. If it wants to vote, there we go. Web links, as well as a couple of videos. And this talk will be on demand by tomorrow, so please bring your customers and coworkers in here. The on-demand tab, so all of our other ones will be in here, and MP Bio will be hanging out here tomorrow. So please feel free to take a look around and hang out. And I'll hand it back to MP, and we'll give them, see if there's any questions in the chat.
Yeah, of course. Before we start answering the question, I do also want to mention that we are running a exclusive promotion with Thomas on Thomas website. So if you are purchasing anything from Thomas, there's a special promotion for the fast prep product along with our adapters. So also feel free to check that out. It is only available through Thomas' website. So that is something that I want to share with everyone, and thank you for the time for participating this online demonstration of the products.
Any questions? I think I saw one in the chat.
Yeah. So we do have one question. Our first one is, there a specific lysine matrix tube that is best for plant tissues? Yes, so we usually will recommend our customers lysine matrix D as star. Lysine matrix D is what we recommend the customer to process most plant tissues. This is the strength of this lysine matrix tube is able to process most plant tissues. Unless, as I mentioned, if it's specifically for seed-related samples, that will be processed, usually recommend for lysine matrix A, as those are much harder tissues to process. But for most plant tissues, more like leaves, roots, those can all be processed by lysine matrix D.
Gavin, I want to add also A, so keeping A and D as your two types of matrices for optimization is best. Yep. When you're just starting up with your tissue. My own experience has been that A actually can provide a little bit more of that grinding that you need for depending on how tough the leaf or the material may be. But yeah, I think A and D would be your best choice to start with.
And Maria, there's a question related to extraction. How can fast prep system and extraction kits help to extract the most DNA from soil samples? Do you want to take that one?
Sure. So the advantage of the fast prep system, as I mentioned before, is in the fact that you can fine tune the amount of sample that you have. If you have soil samples with low biomass and you're required to use more, the different sizes of the tubes and the matrices that we have can help you increase your input and therefore the amount of material you get at the end. Also, to guarantee most DNA, of course, you need to think about the type of soil that you're collecting. Usually it's recommended to collect soil no more than 5 centimeters from the top where your microbes will be. Anything deeper than that, then you may not have as much biomass. So things like considering like the quality of your sample is very important, and then being able to do it in a reproducible way. We have optimized our protocols with the kits and the beads for bacterial, fungi, virus extraction and proper lysis, that you don't have to fine tune that. Usually the programs are already optimized in the fast prep for those sample types of soil or bacterial fungal cells, that if you follow that, usually it will give you consistent and reproducible results. Oh, yep. And I need to mention also with extraction kits that we have the Spin Easy, both the Fast DNA Soil kit and the Spin Easy kits. It's a new spin column line of extraction kits. Has shown great yield purity with these kits. Actually the columns and the reagents are used, the lysis buffer are designed to remove most of the inhibitors are present in soil samples, like the humic acid, heavy metals, and things like that that can interfere with your downstream, to the point that once you have done this extraction in such a short time, you're able to actually use the eluted DNA directly into your downstream application. You don't need to actually do another process of cleanup to remove those impurities. So this is kind of streamlines your process much better, from like, proper lysis to actually being able to remove things in less steps, and then have your DNA ready for your analysis. Is there anything else?
Yes, there's one more question regarding to how can I get the most DNA from diluted water samples.
Yeah, and this is another new feature actually of the SpinEasy kits. We didn't used to have a water kit, but invite you to check out this new addition to our portfolio, because it has really well-designed steps and protocols for handling water samples from low to higher volumes, and that being able to concentrate it on a filter, a vacuum where most of the material, the microbial contaminants are actually captured in that filter. That filter can then be inserted into the lysine matrix tubes with the lysine matrix of your choice. Usually we would recommend the SpinEasy kits also come with beads lysine matrix tubes that are designed to best disrupt bacterial fungal organisms for that particular application for water analysis. And so once you put that in there, start your homogenization, then you get, concentrate your sample, and then therefore get a higher yield. So I think I would strongly recommend that for water analysis. Anything else? Any other questions? Well, I think there's another question. Yeah, maybe Heather can take.
No, it doesn't look like anyone else. Speak out now. All right, I think we don't have any more questions. Thank you all so much for joining us today.
Thank you, Heather.
I hope you have a great rest of your day.
Great, thank you. Thank you for the invitation. Hope you guys let us know. You have our reps that can help, communicate any questions you may have, or anything that you need.
Thank you very much for an outstanding presentation.
Thank you.
Thank you.
Appreciate it. Bye-bye.
Thank you.
Have a good day. Bye.